Hello friends, welcome to BMH Learning. In this video, I'm gonna talk about antimicrobial activity of mint. So, mint exhibits antimicrobial properties. The question is, how can we prove it or how can we check it? So we have a technique for that. First thing we have to do is to prepare mint plant extract. So, we need leaves of this medicinal plant. In order to make the extract, we have to crush the leaves. So, for that we require pestle and mortar. Next we have to add 2 to 3 ml of ethanol to the crushed leaves. Then we have to mix it thoroughly. Next we are going to expose the bacteria to the mint plant extract. So for that purpose, we are using disc diffusion method. So let's prepare the agar solution for this. Let the solution get cool at room temperature. The solution is having a specific type of bacteria called E. coli. We can also take any other bacterial strain in this medium which is sensitive to the used plant, that is mint. But for simplicity, we are only going with one bacteria. The molten agar with the bacteria is layered in a petri dish. The petri dish is then incubated at 25 degrees Celsius. It's because this is the ideal temperature for bacterial growth. After this, we need paper disc. The paper disc is then dipped in the extract made in pestle and mortar. Doing this, we are getting some plant extract on the paper disc. The ethanol soaked by the paper disc is then allowed to evaporate. The disc is then placed on the petri plate. Apart from this extract treated paper disc, we place one more paper disc as a control. It's used for the comparison. This paper disc is only soaked in alcohol. Finally, we leave it for 2 to 3 days and then compare the diameter of the inhibition zone which is formed around the paper disc. Results and interpretation. There is a formation of inhibition zone around the disc which was dipped in mint plant extract. And this shows the plant has antimicrobial properties, thus proving that mint is acting against specific bacteria, that is E. coli. Similarly, other microbial strains can be used to further check the antimicrobial nature of the mint. If the plant being used is having a good antimicrobial property, the diameter of the inhibition zone will be larger. In order to check the minimal inhibitory concentration of the mint extract, different dilutions can be made. The most diluted sample, still showing the inhibition of the growth, would be the minimal inhibitory concentration of the mint. Also, different species of mint plant can be used to get best possible results, as different species would possess varying degree of antimicrobial activity. This was all. Thanks for watching.